University and got a PhD in electrical and electronic engineering. We were very fortunate to have Evren join us in 2003 as a research associate, and Evren's work over this period has been in two areas actually. She started out looking at some problems related to extracting features that would be related to some temporal patterns in temperature profiles in different strains of mice. These were some genetic studies that were going on in the Department of Genetics. And then most recently, in fact, probably for the last 4 years or so, Evren has been leading a computational effort through the Integrated Cancer Biology Program, which is a program supported by NCI in integrating systems control, dynamic modeling, and simulation to achieve a better understanding of cancer, and most importantly, from the efforts that are going on at in the Case School of Engineering in cooperation with the School of Medicine, really trying to understand how to apply these systems and control techniques for therapeutic advantage. So without any further ado, I'll introduce Evren.
Thank you very much. Today we're going to talk about how we can apply dynamical modeling to DNA repair pathways to improve chemotherapeutic gain in cancer. First, I will briefly introduce the repair pathways and their relation to cancer and cancer therapy. Then we will talk about two cancer therapy strategies that are related directly to the repair pathways. And I will introduce the probabilistic cell models that we developed and the mechanistic models of the repair pathways and how we can use these computational models to design optimal treatment strategies for different cancers. Our DNA is damaged through different damaging agents, and for each damaging agent, there are actually targeted pathways, and these repair pathways remove all these damages from DNA, and if these damages are not removed from the DNA, they can accumulate as mutations, and which will lead to cancer. So repair pathways are important to maintain the genome stability. And two of these repair pathways that we're interested in this project are the base excision repair pathway and the mismatch repair pathway, and we will see in detail what these pathways are. So the first pathway we're interested in is the DNA repair mismatch repair pathway, and in short it is called MMR, and it's a multi-protein mechanism, and it corrects the mismatched nucleotides that occur during DNA replication, and this is the only pathway that can actually repair those type of mismatched nucleotides. And it has three sub-processes: recognition, excision, and repair synthesis, and we will go briefly through those three sub-processes. The first process is the recognition, and it is performed by a protein called mutase alpha, and it recognizes the mismatched pairs in the DNA. Here it's a GT mispair, but it can happen in any other pairs. G normally pairs with C in the DNA, but here it paired with T, forming a mismatch, and mutase alpha binds to that mismatch if it recognizes, once it finds the damage. Then once this recognition step is completed, the next step is from here to here, it's the excision step. The rest of the proteins are bound to mutase alpha, and they actually become a huge protein complex, and they excise the DNA. And actually, in mismatch repair process, a long patch of DNA is excised, and you see the excision of this mismatched pair T. Then the last step is the resynthesis of this gapped DNA, and you see that through the mismatch repair system, the GT mispair became a GC again. If mismatch repair system missed this mispair, then you will end up having a mutation in this position. The second pathway that we're interested in is the base excision repair pathway, and base excision repair pathway actually occurs all through the cell cycle, whereas mismatch repair is most dominant in the S phase, where you actually replicate your DNA. And base excision repair pathway is responsible of removing single base damages, and they can happen anywhere or anytime during the cell cycle. And it's very similar to mismatch repair pathway in the sense that you excise the nucleotide, the damaged nucleotide, then the backbone is excised, and then again another DNA polymerase actually resynthesizes that part of the DNA, only the single nucleotide, and you end up with a corrected DNA. And this pathway is called the short patch BER because it only patches the single damaged nucleotide, and it's the main pathway that usually occurs in these type of damages. But for other type of single nucleotide damages, you have the long patch BER. How it is different from this short patch is, instead of resynthesizing only that single nucleotide, another polymerase synthesizes actually a longer patch, and it actually corrects the DNA using this longer patch instead of this single patch. And we will see the mechanistic models of the base excision repair pathway. And when we say mechanistic models, we're actually interested in all the biochemical interactions or all the biochemical reactions happening throughout this pathway for each of the proteins involved in this pathway, both short patch and long patch. We saw that if the DNA damage is not repaired through the repair pathways, it actually ends up accumulating mutations in the DNA, so it is directly related to cancer. And if you look at this table, these are all different repair pathways, but we're interested in the MMR, the mismatch repair pathway, and the phenotype in cancer due to the defects of the mismatch repair is actually the colorectal cancer. And it's the family of colorectal cancers that are called hereditary non-polyposis colon cancers. You won't see the BER in this list because BER is a very well preserved repair pathway, and the defects in BER are not actually causing cancer. And but BER, we will see that BER is very important in the cancer therapy. Repair pathways are very important for also cancer therapy. There are different cancer therapy strategies. Radiotherapy is one of them, and then you have the list of alkylating agents and the inhibitors. These are all the chemotherapy drugs, and most of the damages introduced by all these treatment strategies can be repaired through different repair pathways. Okay? And you will see BER to be one of these pathways. But you will also see that you need mismatch repair in order to mediate the cytotoxicity introduced by these alkylating agents. What it means is that if you are mismatch repair deficient, you will not recognize the damages introduced by these alkylating agents, and you won't respond. The cells won't respond by dying, so they will just go on with their cell cycles. So you won't be able to treat the cancer cells through these chemotherapeutic agent use. So as I mentioned before, chemotherapeutic drugs actually work by producing excessive DNA damage. So the damage is so excessive that the cells decide to die instead of going with their cell cycle. And this is how you actually target the tumor cells, because their replication time is much shorter than they're actually rapidly replicating compared to the normal cells in your body. So this is how chemotherapy actually works. And so the efficacy of chemotherapeutic agents actually depend on the DNA repair capacity, because we have seen that most of the damages introduced by these drugs can be repaired through DNA repair pathways. Okay. But if the damage is too excessive, these repair pathways also signal damage responses, and the cells decide to die. And we will also see that the inhibition of proficient DNA repair pathways are therapeutic strategies in cancer therapy. So the first type of targeted therapy that we're going to talk about is for mismatch repair deficient cancers, because when you lose mismatch repair pathway, you might end up having colorectal cancer, but as well as the cancer phenotype, it also causes resistance to chemotherapeutic agents, as we have seen from the table before. So mismatch repair deficient cells do not recognize the damage introduced by the chemotherapeutic agents, and they accumulate mutations through these damages without actually dying. So you need to introduce a new treatment strategy for such tumors, because chemotherapy is very important in cancer treatment. So if you're resistant to chemotherapy, it's a serious problem. So they have actually used this to their advantage to design a new therapy strategy. They're using halogenated thymidine analogs. These are actually like thymidine in the DNA that T we have seen before. So they can actually be incorporated into the DNA in place of T. And what it does is it actually radiosensitize the mismatch repair deficient tumors, because these tumors are not capable of removing these thymidine analogs from their DNA, whereas the proficient cells can actually remove these and be drug-free. So if you radiate these cells after such treatment, you will actually get more killing in deficient cells, and you will spare the proficient cells. And it's shown by Dr. Kinsella's group. They're the collaborators at CASE in this project, and they have shown that the IUDR incorporation, it's a thymidine analog, in mismatch repair deficient cells are much higher than the mismatch repair proficient cells. So this actually provides differential radiosensitization for these cells. And this is how IUDR works. It's incorporated into DNA after one cell cycle, and it can incorporate it opposite to A, because we know that it's a thymidine analog, so it will pair up with A. And after another cell cycle, this IUDR actually pairs up with G, and they've observed that this is a common mispair in these IUDR-treated cells. So you start with an AT pair, and you end up having a GC or G-IUDR pair after 3 cell cycles. Another scenario is, as I said, IUDR pairs up with G, so instead of a C, it can be incorporated in the DNA opposite to G. So you end up with a GC pair, you start up with a GC pair, and you end up having an AT and A IUDR pair. As I mentioned before, base excision repair is important in cancer therapy. So we will see one example treatment strategy where it can be used to improve the chemotherapeutic response of cancer cell lines. And we have seen that mismatch repair deficient cells are resistant to chemotherapy, and we will see what's the mechanism for that. Here, temozolomide is a methylating agent, and it also works, it's a chemotherapy drug, and it works by creating DNA damages. And most of the damages created are in the form of this right-hand damage, and the rest are the O6-methylguanine. And this damage can be repaired through MGMT. That's a single protein that just removes this damage from the DNA. And if the damages are excessive and if they're not repaired through MGMT, they actually, in the replication, you see this damage, and mismatch repair system will try to repair that damage, but it won't be able to repair it because it's not in the newly synthesized strand. So it will go through a futile cycling, and it will end up signaling cytotoxicity and cell death. But if you're mismatch repair deficient, you actually won't recognize this damage. So this drug won't be cytotoxic for your cells through this left pathway. However, this drug also produces this type of damages, this type of methylation. But although this is 70% of the most of the lesions produced by temozolomide, it is very actively repaired by base excision repair pathway. So base excision repair pathway is very successful in repairing this type of damages. So they won't actually cause cytotoxicity and cell death in tumor cells. But what this treatment strategy does is, if you actually inhibit the base excision repair, then the cells will not be able to repair this type of damage, and it's excessive damage, and they will actually signal cytotoxicity, and the cells will die. So using such a treatment strategy, you can treat mismatch repair deficient tumors. And we will see how, through modeling, we can evaluate the efficiency of this treatment. We have seen that using IUDR together with IR was a treatment strategy, was one treatment strategy for mismatch repair tumors. And our collaborators showed that actually this type of treatment affects the cell cycle dynamics. And that's through these differences in cell cycle dynamics that we're going to introduce an optimized treatment strategy. So we started by modeling the cell cycle. The cell cycle is actually it's a four-state process. The G1 and G2 represent two gaps. In G1, the cells start to duplicate their apart from genetic material, they duplicate the cellular contents. Then they go into S phase where you duplicate your chromosomes, your DNA. And then you go into the second gap phase where you actually check for errors that actually missed the repair processes in the S phase. And if you're error-free, then you go into mitosis and you divide into two cells. So we developed a probabilistic cell cycle model to represent the cell cycle dynamics. It's a finite automaton model, and it has 4 states. Those are the same states as the cell cycle. And we have developed these models on a per cell basis. What it means is that for each cell in a cell population, one of these models will be running, and then you calculate the population behavior by the aggregation of these single cell models. We have represented the jumps from each cell cycle state to the next state using continuous probability distributions, because we know that cells need to spend some time in each cell cycle state before they complete the requirements of that state and before they jump to the next state. So you need to have continuous distributions rather than instantaneous jumps. So we represent all these jumps using probability density functions in this form. From these probability density functions, we will calculate the probability of jump from a given state to the next state. And here actually we represent these densities such that provided that the cell was in a certain state at a certain time, what's the probability for it to jump in another time from that state to the next state. And the parameters of the model are actually the parameters of all these probability density functions of all these 4 jumps in this model. And we have used triangular functions, and we have also used gamma functions. As long as you have 2 parameters to adjust for the place of the mean and the support of that density function, you can use any function you like. But triangular functions are easier to work with computationally. And we have the mean value of that function, and the support, represented by V here, is the parameters for these functions. And those are also the parameters for the model. The output of this model actually gives you the number of cells in each cell cycle state, and from there you can actually calculate the percentages of cells in each cell cycle state. We have first developed synchronous cell cycle models because the first experimental data we got from our collaborators were with synchronized cells. And then we will see how we actually extend this to asynchronous cell populations as asynchronous cell cycle models. So when the cells are synchronized, they all start in G1. And in this modeling effort, we calculate the probabilities of being in each cell cycle state for any given point in time. For the first cell cycle, then we calculate it for the consecutive cell cycles, and we just add them up to calculate the number of cells in each cell cycle state. And I will show you the equations for the first 2 cycles, and it's very straightforward to extend them to any consecutive cell cycle states. So the probability of a cell being in G1 at capital time T in the first cell cycle can be calculated as 1 minus the probability of that cell jumping from G1 to the next state, which is S in the cell cycle, provided that the cell was in G1 at time 0, T0. So it actually spends some time until capital T. And after a certain time, that probability actually becomes 1, and you all jump from G1 to S. And let's represent this by A. This F is actually the density function that is representing the jump from G1 to S. Then you can calculate the probability of cells jumping from S phase to G2, and 1 minus that probability will give you the the probability of a cell being in S state. So here these two F's represent that you were in G1 at T0, then you jumped to S at T1, and you were in S phase at T1, and this is the probability to jump to G2 at T2, but then that would be from the integral would be from T1 to capital T if you're looking for the jump probability, and if you take 1 minus that, you are actually looking at the probability of that cell to stay in S phase. And let's call it B to represent the S phase in first cell cycle. Then you can calculate in a similar manner the probability of being in G2. Then you go to the second cell cycle, and you calculate the probabilities of being each of these states in the second cell cycle. I call them D, E, and F. And you can actually calculate these probabilities for the consecutive cell cycles. So the total number of cells, we represent them as n's in the cell cycle, in the model, sorry. And you can actually calculate them by multiplying these probabilities to the initial number of cells in G1. And you just add them up for the first cell cycle and the second cell cycle. And you will notice that we have a factor 2 here. It actually represents cell doubling after the first cell cycle. Okay. So the cells became double in the second cell cycle. This first model does not include cell death, but we will see that the most general model we have developed includes cell death. So we will account for cell death. Then from these numbers, you can get the percentages. We need the percentages because that's the experimental data we get from our collaborators. We estimate the parameters of the model using flow cytometry measurements. From the flow cytometry measurements, we get the percentages of cells in each cell cycle state. And actually, in the flow cytometry measurements, the G2 and M phases are measured together. So we actually lump those two states together. So we have 3 states in our model. So 3 states would mean 6 parameters, 2 for each of the state jumps of that probability density functions that I showed before. And we have used the sum of square cost function. There's only one constraint on the parameters of these probability density functions, and that is density functions should have 0 values for negative time. Since most of the experiments are done on asynchronous cell populations, we needed to extend our model to asynchronous cases. And this is the most general model because it is asynchronous and it also involves cell death. So associated to each state in the cell cycle, there is this cell death state. So the cells actually can either decide to continue with their cell cycle or can die from that state. And all these jumps are again represented by similar probability density functions. And here the states of this cell cycle model are defined as small n's. Those are actually the flows from or to each cell cycle state per unit time, flow of cells per unit time. And capital N's are the same as the previous model. They represent the number of cells in each cell cycle state. These are the equations how you can calculate those flows. Again, notice that the flows going into G1 will be doubled because the cells are coming out of G2N phase and they actually become 2 cells after mitosis. And in order to be able to calculate these flows in the asynchronous model, you need to know the initial flows. And we're going to actually calculate these flows from experimental data for the cell populations that are at steady state. So these are usually the control cases in each experiment. Then you can calculate the number of cells in each cell cycle state by just integrating those flows and subtracting the cells that are going to the next state and that are also going to the cell death state. We are currently applying these probabilistic cell cycle models to 3 different problems. The first data set comes from Dr. Kinsel's lab at Case. And they're interested in the differences in the cell cycle profiles of mismatch repair proficient and deficient cells under IUDR under different treatment conditions. One treatment strategy is the use of IUDR, which is a halogenated thymidine equivalent, and followed by the ionizing radiation therapy. And they're actually interested in how these treatments affect the cell cycle dynamics. And the second data set comes from Dr. Laufenberger's and Dr. Sampson's labs at MIT, and they're interested in the alkylating agent MNNG. It's again a chemotherapeutic agent, and they're interested in again the cell cycle dynamic differences in TK6 cells before and after treatment. TK6 cells are human lymphoblast cells. They're actually bone marrow cells, and they also want to know how this drug actually induces cell cycle arrest and cell death mechanisms, because they don't know from which cell cycle state the cells are dying most. And so we will use our models to actually come up with different scenarios of cell death. And they will go back and experimentally verify the findings of the model. And the last data set again comes from MIT, and they're interested in U2OS cells, and the U2OS cells are again bone cancer cells, and they're interested in the effect of ionizing radiation on these cells, on the cell cycle dynamics, and they're actually seeing permanent and temporary arrest, and we're going to study these mechanisms through the cell cycle models. So the first data set we have used came from mismatch repair deficient and proficient cells. Those are the HCT116 cells, and they were treated by IUDR. And the aim of this study was to see the cell cycle dynamic differences under the treatment under only IUDR treatment conditions. The second set of cells were also synchronized mismatch repair proficient and deficient cells, and this time IUDR treatment was combined with followed by IR treatment. And by the time of IR, the cells were already asynchronous. So in the second case, we only used asynchronous cells, cell cycle model. And in the first case, we used the synchronous cell cycle model. And for the asynchronous cell cycle model, we just got the initial flows through these synchronous cell cycle models. And in addition to the flow cytometry measurements for the cell cycle distribution data, we also get IUDR incorporation measurements from our collaborators. And we are developing the models for this incorporation, and we will see how we're going to use it to optimize the therapy. And these are the modeling results for the first set of data. The data is actually shown in solid lines. If it's not pattern as the data, then here the triangles actually represent the G1 phase, the diamonds are the S phase, and the squares are the G2 phase. And you can see that the models actually captured, these are the modeling results, captured the experimental, the trend in the experimental data pretty well. Okay. And we have applied these models to deficient cells with no treatment, proficient cells with no treatment, and then deficient and proficient cells under IUDR treatment conditions. And this work was actually published in Cancer Research if you're interested in reading it. And these were the parameters that we got for those 4 cell cycle models. And when you compare the parameters, you can actually see the dynamical changes in these cell cycle profiles of these proficient and deficient cells.
What about AMs and
Again, the AMs are the parameters of the models were the means and the support values of the probability density functions that represent actually the probability of jump from one state to the next state.
Evan,
Yes.
how close of your model compare with the realistic data?
How close? Okay.
Yeah.
Well,
Do you have
I mean, I don't have the error measures in the slide, but I mean, you can actually, this solid line is the true S phase, and these diamonds are the S phase from the model. Right. So it's very close. Then we got the second set of data from our collaborators, and that one was IUDR treatment followed by IR treatment. And we're interested in seeing how treating the cells with IR after IUDR actually affects their survival compared to only IUDR treated cells or no treated cells with only IR treatment. And this was a longer experimental data compared to the previous data. So our synchronous model used in the treatment cases or no treatment cases with IUDR should actually match, but we have seen that for longer data sets, for the later times, the model did not match. So we went back and checked what the problem was, and we have seen that there's a biological reason behind it. The cells were synchronized through starvation. So starvation induces a cell stress on these cells, and once they start their first cell cycle, they're actually slower to recover from that stress. And then in the second cell cycle, they're actually much faster. So in order to address this problem, what we did is we have used 2 different parameter sets for the first and second cell cycle. And the model actually matched the longer experimental data as well as the shorter data previously. And we have used asynchronous models for the cases where the cells were treated by IR and IUDR plus IR, because the cells are already asynchronous for when the IR was applied. And we are using the synchronous models with no treatment and IUDR treatment to obtain the initial conditions for these asynchronous models. And currently, we have finished developing the models for mismatch repair deficient cells with IR treatment, and we're working on developing the models for deficient cells with IUDR plus IR treatment, and for proficient cells with IR only and IUDR plus IR treatment. And I'll show you a series of results of our synchronous model for longer data with 2 parameter sets. So we see that we have a set of parameters for the first cell cycle, and we have a second set of parameters for the second cell cycle. And here the blue lines are the G1 measurements and the model, and the green represent S phase and red represents the G2 phase. And the circles are the experimental data, and the stars are the model fit. And you can see that it was actually a close fit to the experimental data. Yes?
So did you fit the model to this experimental data?
Yes.
Or the model parameters were chosen prior to getting the data?
No, I fit the model to this experimental data.
Okay, so this is showing that you're able to fit the model.
Yes, but we're going to use those models. Once you fit the models, we're going to use them actually to estimate further times.
Right.
Okay. And we also, I don't have the graphs here, but we had multiple measurements for this first period of the data, and multiple measurements of these cell cycle distributions, and the model actually fits pretty well without actually, I mean, the model validates pretty well with those experimental data. And this one was the deficient cells, MMR minus cells, under no treatment conditions for longer data. And this is for deficient cells with IUDR treatment, and you can see that model is pretty successful in fitting to the experimental data. And actually from the experimental data, you cannot really get the means and the variances. You only get the distribution. So what the model provides you is actually the details in these dynamics. So you can actually see what's the variance and variability in that state, and what's the mean value in that state. And you can compare the how these parameters change after treatment. Then this is the modeling results for the proficient cells with no treatment conditions, and you can see that the model is pretty good in following the trend in the experimental data. And this is the modeling result for the proficient cells with IUDR treatment. And we also use the parameters of the model to actually compare the dynamical changes between proficient and deficient cells under different treatment conditions. Then we first applied our asynchronous model to deficient cells treated with only IR, and currently we are also applying it to deficient cells treated with IUDR followed by IR. IR is the ionizing radiation. And the first one is the synchronous model we have seen before. And we use this, we get the initial flows to initialize our asynchronous model from this model using the parameters from this model. And these are the three this is actually the same model, but the IR is applied in 13 hours, 16 hours, and 21 hours. And we use those three data sets together to estimate the parameters for IR treatment. And you can see that the model is actually successful in generating the trend after IR treatment for these deficient tumor cells. And these are the parameters for this set. And you can actually see that the mean value in G2 is pretty high. That actually corresponds to the biological fact that the cells are G2 arrested after IR treatment. Okay? And you can actually see that reflecting in the parameters of the model. So how can we use our computational models in designing optimal treatment for these mismatch repair deficient tumor cells? The aim is to find the optimum time to irradiate the IUDR treated cells so that you actually maximize the cell death in deficient cells while you're minimizing the cell death in proficient cells. And the biological facts we use in designing such an optimal treatment strategy is that the incorporation of IUDR, the incorporation of this drug to proficient and deficient cells are different. And we want to actually find the time where this difference is maximum. And you want, of course, more incorporation into deficient cells, so you get more cell death in deficient cells when you apply IR. And another biological fact that we know is that cells are most sensitive to IR in G2 state, and they're less sensitive to IR in S phase. So we're going to obtain a prediction of the distribution of cells in each cell cycle state from the cell cycle models that we developed. And in parallel, we're also developing IUDR incorporation models, and we're going to actually obtain the incorporation profiles from the IUDR incorporation models. And we will actually find the time when the difference between IUDR incorporations are maximum, and the mismatch repair deficient cells are mostly in G2 phase, and mismatch repair proficient cells are mostly in S phase. So if you can actually radiate those cells, at that time you will get the maximum killing. Okay. And the second set of data came from MIT, and they're looking at the effects of MNNG, the alkylating agents, on TK6 cells. And they're looking actually at the differences between different doses of MNNG, low dose and high dose, and its effect on cells. And they're not looking this problem through the treatment strategy point of view, but they're looking at it from basic science point of view. So they're interested in the mechanism of cell death and the mechanism of cell cycle arrest in these cells when they're damaged by this agent. The cells are asynchronous in this case because they cannot synchronize TK6 cells through starvation or chemical inhibitors because they die. So there's no way to synchronize those cells. So we cannot use synchronous models in this case. But for the control case, in order to be able to get the parameters for the control case, when I say control, it's the untreated case, you cannot actually use the cell cycle models because the cells are not synchronized. If they can be synchronized, you can use the synchronous cell cycle models to obtain the parameters. But asynchronous cells are actually at steady state. So the cell cycle profiles are flat. So you don't get any information out of the cell cycle profiles. So you need to actually disturb the steady state. And in this case, they've used centrifugal iteration technique. What it does is it actually spins the cell population. And the lighter the cells, they're going to be going toward the outer sides of the iteration. So you actually collect the flow here. So the first flow coming out of this machine contains most of the G1 cells because they're lighter, because they haven't replicated their DNA content yet. And as time goes on, the flows get richer in S-phase cells and then finally G2 cells. So they've actually collected 7 different flows from this iteration for their TK6 cells. And we have used 6 of these different flows to do the parameter estimation. And we have used the 7th set, which was a longer time course to validate the model. And these were the model parameters, and our collaborators said that they make the most biological sense from their biological insights. And this is the model fit. Again, the circles are the experimental data, and the stars are the modeling data. And we're going to use, why did we need the control case parameters? Because we're going to use these parameters to generate the initial conditions for the asynchronous model that is going to evaluate the effect of the treatment on these cells. And we're currently working on the low-dose MNNG treatment on these TK6 cells and its effect on cell cycle. Next, we will move into the mechanistic models of base excision repair. And this is a model from the literature, and it involves all the proteins that we have discussed before in the base excision repair pathway. And it actually models the biochemical reactions of these proteins together with the DNA. And these are the sets of equations that define that model. They're actually differential equations. And the biological kinetics that are used in this case are Michaelis-Menten kinetics. And you can actually use mass action kinetics instead of this specific Michaelis-Menten kinetics. And we've actually taken that model from the literature, but we modified it because we're interested in the inhibition of base excision repair pathway. And in this case, we're also interested in a different glycosylase than they have used in that model. And we have included these inhibition dynamics to our model to study the effect of inhibition on different BER pathway with different BER capacity. We will see what we mean by that. And these are the equations defining that model. They're again ordinary differential equations. For most of the enzymes, we have taken the parameters from the existing literature from where the model was published first. But for the methoxamine parameters, we have actually searched the literature for experimental data. And we have found one paper from 1985, and we have estimated the parameters for methoxamine and used it in the model. And we know that the inhibition of our collaborators told us that the inhibition of base excision repair actually depends on the BER capacity of each cell. So BER capacity is not the same in each and every cell or between between different individuals where the cell comes from. So in order to capture that biological variability on the efficiency of BER process, we have run Monte Carlo simulations. And in these simulations, what we changed were the enzyme concentrations. You can as well change the enzyme constants, but we kept them constant. And we have changed enzyme concentrations because not every individual cell would have the same enzyme concentrations at any given time. And we have clustered the outputs of these Monte Carlo simulations into 3 classes. And we named them as highly efficient base excision repair, medium efficient base excision repair, and inefficient base excision repair. And we wanted to see the effect of inhibition by methoxamine on these 3 different classes of base excision repair capacity. And we have seen that actually methoxamine can inhibit the medium efficient base excision repair most, whereas it is least effective on the high efficient BER. And I will show you just the 3 clusters we got from the Monte Carlo simulations and through clustering. And here you see the effect of inhibition. The dotted lines are actually the inhibited base excision repair output. And this is the high, blue corresponds to the highly efficient BER, red is the medium efficient BER, and green is the least efficient BER. And we have seen that inhibition of BER pathway is an important treatment strategy for the mismatch repair deficient tumors because they are chemotherapy resistant, but chemotherapy is the most effective treatment on cancer. So you really want to use chemotherapy on these tumors. And BER, inhibition of BER is one way to be able to use chemotherapy with these tumors. And efficiency of this inhibition, of course, plays an important role in the therapeutic efficacy of the cancer drugs. In this kind of treatment strategy, you actually want to find the dose of the original drug and the dose of the methoxamine that is the inhibitor for this repair pathway, and also the timing, how to apply the combined treatment. And through the use of mathematical models, you can actually look at the dynamics of the BER pathway without the treatment and its efficiency and its capacity. Then you look at the inhibition of the BER pathway through methoxamine, and then you look at the efficiency of this treatment analytically. Then you can actually introduce the original damage induced by the chemotherapy agents and how the inhibition will actually affect its repair. Because when we say inhibition, it's not 100% inhibition. Methoxamine can inhibit to a certain extent, so you can only, even the dose is high, it won't actually inhibit the base excision repair 100%. So you need to actually study its inhibition effect, and you can only give so much dose of this drug to patients. So you need to know what's optimal for actually for each patient. So these are a list of future projects that we're working on. Some of them we've already started to work on. Similar to the mechanistic model of the base excision repair model, we want to develop a biochemical mechanistic model for the mismatch repair process, and it's going to be a stochastic model. And we're specifically interested in using this model for IUDR processing, capacity of mismatch repair pathway. And the experiments to parameterize these models are already being carried out at our collaborator Dr. Kinsella's lab with a purified protein system, because you need to purify the proteins in order to obtain their biochemical reaction rates. And all of the proteins are now purified, and we're starting with the actual kinetic assays of these proteins. And of course, in order to be able to use these models at the molecular level, to say something about at the organism level, you need to go through different levels. You need to extend this modeling and experimental scheme to cellular systems first, to go from there to in vivo systems. And that is what we're planning to do in the next couple of years. And as I said, this is crucial for the translational research. And we need to, of course, include cell death pathways to our model, because we're looking at survival or cell death in tumor cells and in normal cells, so it's a natural extension to involve cell death pathways. And they're currently studied experimental in Dr. Kinsella's lab, both from apoptosis and autophagy cell death pathways point of view. And we will couple our models to the models of the cell cycle dynamics to cell death pathways and study the cell death profiles under different treatment conditions. And we're interested in different chemotherapeutic agents, not only IEDR or temozolomide, but we're interested in a class of chemotherapeutic agents. And one such agent is 6-TG, and the experiments are currently carried out in Dr. Kinsella's lab. And once we get the data, we will start modeling these aspects of the treatment. So, and of course, I would like to thank my mentor for Dr. Lapara. And this was a great project, so I'm really happy to be part of it. And I would like to thank all our collaborators for their data and also for biological insights and biological training. And this was a part of ICBP, the Integrative Cancer Biology program at CASE, and it is funded through NCI. So thank you very much.
Thank you. We've got time for a few questions. Any questions? I've had 4 years or more to ask mine, so.
I had a question.
Sure.
So you talked about the synchronous model, and then you switched over to the asynchronous model, and I didn't really see derivation of the asynchronous equations. Is it just really a very simple process of extending it to an asynchronous model?
It is actually what we needed to do, though. We needed to redefine the states. It's the same model.
The derivation, or
I just show the equations. Let me show you the equations. So, but it's the same model. Anyway, it is the same model. It's just the state definitions are different because you don't know the initial distribution. You don't know how. To be able to calculate the probability of jump from one state to the other, you need to know how much time the cell spent in each cell cycle state. But with the synchronous model, you know it because they all start at time 0 in G1. But for the asynchronous model, you have no idea how much they spend. But you actually have, not analytically, but numerically, we've calculated the steady state form of these models. So from that distribution, from the steady state distribution, we get the initial distribution of cells in each cell cycle state. But the model is, the principle of the model is the same. So let me actually see. Okay, so these were the equations defining the states of that asynchronous model. So those are actually, what you're doing is you're taking the flow into one state, and you're actually come over with the density function to determine the output from that state, the output flow from that state. So the idea is the same as the synchronous models.
It just seems really fascinating that you can take a synchronous model and then just easily transfer it over to an asynchronous model. Because there are so many, just seems like there's so many applications for doing that. Okay, thanks.
Anything else? Okay. Thanks again, everyone.
Thank you very much.